Do I spy a post about molecular cloning? Why, indeed, I do. Molecular cloning is one of the most important techniques that you'll use in biochemistry, so let's take a look. About like making copies of your dog or anything. What we're talking about is just taking a gene from one place and sticking it into a different place that's easier to work with. And so we call this like recombinant DNA or recombination because we're combining DNA from different places. So usually what we do in molecular cloning is we take a gene containing the instructions for a protein that we want to study. And then we stick that gene inside of this thing called a plasmid vector. And so this plasmid is the circular piece of DNA that we can stick into bacteria. And then the bacteria will host this plasmid like outside of their own DNA. So the bacteria have their own DNA, and then they'll host this extra plasmid. So these plasmids actually come from bacteriophages, which are bacteria infecting viruses. And we just kind of like repurpose them to serve as vectors or vehicles for our gene of interest. And as I'll get into later, it's not technically the gene, it's the cDNA, which is like an edited version of the gene. It has the regulatory information removed. So we take this plasmid and it has the recipes for making that protein, and then we stick it into the bacterial cells. And we call this, when we stick it in, we call this process transformation. In addition to the gene or the instructions that you're interested in, the plasmid also has a selection marker. So usually this is an antibiotic resistance gene. So bacteria that take in the plasmid, so not all the bacteria will take it in, but those that do, they'll have the antibiotic resistance gene. So they'll make that antibiotic resistance protein. So if you grow the bacteria in the presence of that antibiotic, so basically you spike the bacterial food with the antibiotic, then only bacteria that have the plasmid will be able to grow. And so the bacteria, this allows the bacteria to multiply, but only the ones that contain your the plasmid. And so this will multiply and they'll make lots and lots and lots of copies of that vector. Problem is, not all of the plasmid necessarily has your insert in it. So we take steps to ensure that, or to try to ensure that only the plasmid that actually has your insert actually gets in. But sometimes like the parent vector, so basically the unmodified plasmid, can get in there. And this will also be able to grow because it also has the antibiotic resistance gene. So what we need to do is we need to make sure that we choose bacteria that actually have our gene of interest. So what we do, we do the transformation. So usually what we do is we do this with heat shock. So basically we take weakened bacterial cells and that are made chemically competent. So basically what this means is we weaken them so that when we use heat shock, so we stick them in a warm water bath for like 40 seconds, pores will open up in the surface of the bacteria. And if you do that well, after you've drenched the bacteria in this plasmid, with this plasmid vector you made, then all of that plasmid will be able to go into the bacteria. And then you do that with like a solution of the bacteria in a tube, and then you plate it. So often you do this with like a spreader or with like glass beads that you shake around because you want to make sure that the bacterial solution gets all over the plate. And so when I'm talking about plates, so these are like agar plates. So agar is a sugar that we can use to make a gel, And in the gel, we make it so that it's like made of usually LB, lysogeny broth. And so this is like bacteria food. And so we have the LB and we spike it with the antibiotic. And then we do that while the gel is like all liquidy, and then we pour it into this plate and it hardens, and we have this gel that the bacteria can grow on. So we take our bacteria that hopefully have the plasmid, and then we plate it on these plates, and we let the bacteria grow in an incubator. So we just basically keep it warm. And you want to make sure that you put the plates in upside down, so that condensation doesn't form on the top and then drop down and mess things up. So usually it takes, depending on the strain or whatever, like it'll take about a day. So you put it in the incubator and then you come back the next day and you should see a bunch of little dots, little globby dots. So these are called colonies. And so basically each colony represents a bacteria that's taken in the plasmid and then multiplied. So when you might think, like, so when you originally spread all that bacteria all over, you think, if you didn't have antibiotics, what you'd get would be like a big lawn. So basically it'd just be like covered in these bacteria. But instead what happens is that only some of the bacteria actually take in the plasmid, and only some of the plasmid actually has your insert inserted. So any that take in the plasmid, and most of the bacteria actually probably die, because the chemically competent cells are basically, we weaken them to the point that they're almost dead, and then we transform them. So yeah, we actually usually have to do like a recovery step after we transform them. So we let them grow a little bit in the tube in the absence of the antibiotic before we even try plating them. So what happens is that you only end up with like a few of the plasmid containing bacteria, and they're kind of like scattered all over the plate as long as you did the spreading out well. So you want to, when you're using the glass beads and stuff, you want to make sure that you don't have too much and that you actually do it till it like dries, or else you'll get a bunch of colonies like around the rim of the plate instead of spread all over. But basically you get these globby dots, and each of these globby dots represents a single bacteria originally that then multiplied and multiplied and multiplied and multiplied on top of itself, so you get this glob. And each bacteria in the glob should be genetically identical, barring any mutations or whatever that occur during the copying process. So that means that one colony might have just the plasmid, the original plasmid. One colony, most of the colonies should hopefully have the insert in the plasmid, and some of the colonies will have the plasmid with the insert, but the insert will be messed up. Like there's some mutation that happened, or it got like there was a problem during either the cloning step or the insertion step, and so there's going to be something off with it. So basically you want to, before you go on to work with the plasmid, you want to make sure that it actually is the one you want. So what you'll do is you'll pick a few of these colonies, depending on if you're having a bunch of trouble with the cloning or whatever, you might do more colonies. If things are going easy, you might just start with like 3 or so. And what you're gonna do is you're actually gonna take your like pipette tip and dip it in the colony, and then stick it in a tube with about 5 milliliters of the LB broth, which is the same thing that you made with the plates, but here you don't have the agar. So what you're doing is you're growing it in suspension. So you grow it in these tubes, let the bacteria multiply and multiply and multiply, and now so basically the goal of that part is just to get lots and lots of copies of that single clone. And now what you want to do is get those copies out. So you can use a process called a mini prep, which is like a kind of proprietary term, I guess, but basically for alkaline lysis. So lysis means like break open. Alkaline refers to basic, as in like the opposite of acidic. So basically the idea here is that you break open the bacteria and then you're going to purify out the copies. So this is done with like these little spin columns usually. I have a post on those too that you can look at if you're interested. So when you do this, the purification process takes advantage of the fact that the bacterial chromosomes, the bacteria's own DNA, is a lot bigger, and so it's not going to be able to like recover itself kind of during the purification process, so it's just going to clump up, and so you won't have to worry about it, and so you should just end up with the pure plasmid once you purify it out. So now you have your plasmid, but you have two questions. Is it in there? And is it actually right? So there are a couple ways if it's in here. So one of them is analytical restriction digest. So basically you take a restriction enzyme, which is an enzyme that recognizes specific DNA sequences and cuts them. And so if you choose, you can use that to see if you try to cut the plasmid and see if it gets cut, and how many pieces it is. So if you choose like a cut site that's only in the insert and not in the plasmid, then only if the plasmid has the insert will it get cut. And so then you can use you can use agarose gel electrophoresis to separate the fragments by size and see if the sizes are what you would expect if it has a certain number of cuts. Another way is a colony PCR. So this is basically instead of cutting the plasmid, you make lots of copies of a region of the plasmid, and in such a way that the size of the products or the presence of the products will depend on whether the insert is there. So you can like use primers. So primers are primers are like short pieces of DNA that you use to bookend the region that you want to be copied. And they serve as like start sites for DNA polymerase, the copier, to go between. And so if you have a site that's only in the insert and not in the plasmid, then you'll only get the product if the insert is present, and then if you choose ones that are just on the plasmid, you'll get a different sizes if the insert is there or not, if you like do it so that it overlaps the insert. And I have posts on these as well. So a good thing about that method, so colony PCR we call that, is that you can do it without having to actually purify the plasmid out first. So at the same time you're picking colonies to grow overnight, you can do take that pipette and take a little bit of it and just mix it in the liquid buffer already, and just do a PCR directly on it. So with the PCR, it'll break open the cells, and then if the DNA is there, it'll copy it. So the good thing about that is you don't have to purify it, and if you keep track of which colony corresponds to which growth, then you'll know when you so you don't waste your time doing mini preps on ones that didn't work or whatever. But either of those methods won't tell you if all the spelling is right. And so for that, you usually send it for DNA sequencing. So this is like Sanger sequencing. I'm gonna ship it off to a place like GeneWiz, or I don't remember what company we used to use, because I know we switched. Some schools often have their own sequencing cores that'll do it for you. And basically the idea there is that you use PCR primers that at the start and end of your gene, kind of, so that you can make sure all of the spelling is correct. And so I have a post on how to analyze that stuff as well. So yeah, so basically just a little more detail. I'm not going to go over all of this. I just didn't have time to like make slides or anything. So this is just some graphics I have. The key thing to take away is what I was talking about before, is the idea that the DNA version that you put in isn't like the gene, it's the cDNA. So cDNA stands for complementary DNA, and it's the DNA version of the messenger RNA. And so basically what happens in cells is that genes make, so the original recipe for a protein is in the gene, which is in the DNA, and then an RNA copy of that is made and processed into mature RNA, mature messenger RNA, mRNA. And so that maturation process involves removing regions called introns, which are between the protein content containing exons. And so basically the introns are like regulatory regions, and so they get removed. And so it's the DNA version of that mRNA that you're going to stick into your plasmid, because the bacteria aren't going to be able to do like all that processing, that splicing out of those introns. And if you leave the introns in there, then the ribosomes, so those are the protein making complexes that are going to travel along the mRNA to make the protein, they're going to, if you have the introns in there, they're going to make gibberish. And so you don't want that to happen. So you use the cDNA. But normally we just talk about like sticking the gene in, but I just wanted to introduce like these technical terms so you don't get confused about. And so we have different types of vectors that we can use for different types of cells. And yeah. And so a couple of the key ways that you can use to do molecular cloning, so to actually get your insert into the plasmid. So I'll probably do further posts. I mean, I have written posts on these, and I'll probably do more detailed videos on them at some point. But a key, the original way is restriction cloning, where you use restriction enzymes like those DNA cutters we talked about before. But in this case, you're using them to cut around the insert from where it's originally, and then put it into the plasmid that you want. Another way that is what I use usually is SLIC, sequence and ligation independent cloning. And the idea with this is that you use PCR to make copies of the insert out of its original place. Instead of like cutting it out, you just make lots of copies of it, and then you make lots of copies of the part of the plasmid that you want, and then you have it so that they have ends that are the same, and then they get recombined together in the bacteria cells. So yeah, so the other thing is that you want to, another thing about the idea of some of the parent vector or the parent plasmid still being in the sample, I mean, like in the colonies that you have. And so there are a couple ways that you show this. So when you're doing restriction cloning, normally like you purify the vector DNA, and you purify the insert using like gel purification. So basically you run an agarose gel to separate them by size, and then you like extract it out of the gel. So you shouldn't have the any parent plasmid in there, because you should have only had like the cut version be purified, but it can still happen. But when you're doing a PCR based method like SLIC, you have you don't have any sort of purification. What you can do is use DPN1 digest. So basically, in this step, the temp, so basically your new version is going to be PCR copied, and the template is not PCR copied. And the importance of this is that when when plasmids get copied in bacteria, they normally get like methylated in the cells, and this makes them vulnerable to a restriction enzyme called DPN1. But the new version that's PCR copied, it doesn't get methylated, and so it doesn't get cut, and so it'll be fine if you DPN1 digest it. So this should hopefully get rid of the parents. Yeah, that's so one thing that you can do to prevent with restriction enzyme based cloning is that if you have like sticky ends for the plasmid, you might need to dephosphorylate the ends so that they can't join. And I have more on this in the other post, but I just want to introduce it to you here so that you know that I have posts on that and that it's something that you might need to be worried about, but I'm not going to go into detail now. When you do that transformation, you want to have a couple controls. So with the case of restriction cloning, you want to have like an uncut vector, so this is your positive control, so that'll make sure that the transformation process worked, so that plasmid actually got into the cells. So on this plate you should have lots of colonies. And then with the cut vector, so with the cut vector, you should have, this is like your negative control, that you don't have any parent or self-circularized vector. And it also shows that the antibiotics were effective. So that's basically all for today.